Hello. Hello.
Yeah, hi.
Um,
Thank you, ma'am, and I'll hand over to you. You may share your slides and start.
Great. Okay, thank you. I'm going to, let's see, get this sharing. Hello, everyone. I caught the last couple presentations, some interesting projects you're working on. And I'll be excited to hear how things end up by the end of the week. So today, we're going to be discussing design and data considerations. And there's a number of different topics that we're lumping into this one overarching theme. So I'll jump right in. So first off, all of the topics that I'm going to discuss today, I will go over, do an overview, and then I'm going to apply one example from one trial. So if you'll recall yesterday, I mentioned that I work on a lot of cryptococcal meningitis trials. And my team in the last couple of years has also started moving into tuberculous meningitis. And so one of our studies that we just wrapped up at the beginning of this year was called Harvest, where we were looking at high dose oral rifampicin to improve survival for adult tuberculous meningitis. So this was a double-blinded, randomized, placebo-controlled phase 3 trial. We had a population of about 500 adults with TBM, which has a very high mortality. And we were looking to see if a high dose oral rifampicin could improve 6-month mortality. So everything I go over today, I'm going to reference this trial as the example. So we'll start off with recruitment of participants, screening. So to begin, you're first making contact with potential participants. Many times in a clinical trial, this is patients in a clinical setting. This could also be contacts via community partners or marketed outreach. Most of the time, I would say many RCTs are conducted in the clinical setting. But my COVID hydroxychloroquine trial example from yesterday, that was marketed outreach. We basically like blasted a link all over Twitter saying this is please take this survey, screen yourselves for this trial. So first thing is making contact. And then the next thing is determining criteria. So the participants in the trial are going to be a representative sample of the larger population that might benefit from the results of the trial. So the criteria to the participant is informed by the overall research question, right? So we have the entire population, but for our study, we have a target population. And the key features of that target population are the inclusion criteria. And then the study population is then also has exclusion criteria, which are the features of potential study participants who might meet the inclusion criteria, but present with additional characteristics that could interfere with the success of the study or increase the risk for unfavorable outcome. So in my example of the Harvest trial, which was looking at high dose rifampicin as a potential intervention for tuberculous meningitis, we have some pretty typical inclusion criteria. This must be your first episode of TBM. You have to be an adult, 18 years or older, and you have to provide informed consent, which we'll cover in a second. But exclusion criteria, this includes things like a known allergy to many of the drugs used to combat tuberculosis. This includes any other steroid, some contraindicated medications like corticosteroids. Even something like the participant is not likely to attend regular visits. That's something, so the contraindicated medication, that's something that would maybe lead the patient, the participant, to have a unfavorable outcome in the study. But something like exclusion criteria number 7, unlikely to attend regular clinic visits, that's an exclusion criteria to protect the study itself. We want to have the least amount of missing data. We want participants who show up regularly to all of their scheduled visits. So those are some examples. Informed consent is a very important part of clinical trials. It means that the participant completely understands a number of things. The purpose of the trial. Why is this trial being conducted? Why am I being involved? The procedures involved. So the fact that they're going to get randomized, they're going to potentially get an intervention, or they might get a placebo. They need to be aware that they have an equal chance of being randomized to both arms. They need to know potential, in addition to randomization, they also need to know other procedures involved. Are they ever going to have a lumbar puncture? Are they ever going to have to discuss their families, for example, their household income, any other sensitive topics? Are they ever going to have to do anything else during the study? Then potential benefits and risks, right? Describing this is what we hope to see in our trial from our intervention. And we also could encounter some adverse events, and the participant needs to be aware. They also need to understand that they are able to stop participation at any time. This is a requirement of informed consent of any trial. The participant is always a volunteer and can withdraw consent to participate at any time. So this informed consent is essential for ethical research. That's that word that we said was going to come up a lot throughout this workshop, is the ethical research. And I will point out that there are, depending on the study design, there are some times when consent could be waived, right? So an IRB, an institutional review board, might waive or alter the requirement of informed consent if the research involves no more than minimal risk to the subject, if the waiver or alteration is not adversely affected the rights or the welfare of the subject, especially in the case where research could not practicably be carried out without the waiver or alteration. I think there are some experiments that are conducted in the field of psychology that they have to design the study in a way where the participant doesn't know they're being in the study, but those are particularly controlled. And again, the study is designed in a particular way. So that's informed consent. So in the example of the Harvest trial, you'll recall that our participants are patients who are suffering from tuberculous meningitis. They have an infection of their brainstem, which is very often affecting their ability, their consciousness. A lot of them have debilitating headaches. They have a Glasgow Coma Scale score of less than 15. They're not able to provide their own informed consent. And in our case, we also allowed for a legally acceptable surrogate. So a family member, somebody who brought the patient to the clinic, that person understands the trial completely, signs on behalf of the participant. And in our case, if the participant survives long enough and is able to recover to the point that they are conscious and able to provide their own consent, we actually re-consent them later in the trial so that we have that, buy-in continually for participation. Okay, so at this point, before I move on to the next section, I'm going to stop and ask if anybody has questions regarding screening, recruitment of patients, exclusion criteria, informed consent. Any questions?
I'm in trials where the participant is paid a certain amount. There the withdrawal of voluntary withdrawal at any point of time, isn't there a conflict of interest out there? Because there may be a reason why the participant choose to come into the trial because of financial incentive, even though they may not admit it. So truly a withdrawal from their, there's a kind of interference with respect to the voluntary nature of it. Any comments, ma'am?
Good question. And other panelists, please feel free to respond as well. From my understanding, a participant is a volunteer, even if they're being paid. A lot of times paying a participant is a way to help the study a way to help the participants and a way for the study to benefit, to incentivize the participant to keep showing up. So if a participant wants to withdraw and stop consenting, they're welcome to do that at any time. They just won't continue to get paid. Does that answer your question?
Also that, I mean, incentivizing is something, as she said, that you want for the participant to continue to participate or to attend visits, etc. You don't want to give so much that it is coercive, and now they're actually joining not because they are really consenting, but they are being almost bribed to join. Who determines the amount? That is, your ethics board will review that and say, no, the amount you're offering is actually the incentive that is a coercive incentive. And so the amount usually in most trials that I've been participating in is just to defray costs. Like if you have to take transportation to come to the clinic, they will pay for the transportation, they will pay for a meal afterwards, but it's not giving the participant lots of money. This is not a business for them to participate in trials and make money and live off of that. But the amount and the the frequency, etc., that's something that your ethics board will actually see if it's coercive or not.
Thank you. Great question. Great. Okay. So next up, I'd like to talk about assignment to intervention arms. So we're talking about randomized clinical trials. So why are we randomizing? In intervention arms, when we randomize, both arms have similar distributions of types of participants. We want to control as much as we can about these participants. And randomizing allows us to have, if we have a large enough sample, we're going to have a similar types of participants in both arms. So that's controlling that. Similarly, for the participant size, they have the same probability of either intervention, right? We don't want to bias anything. And participants for them, it's the ability to have the same chance of the intervention or the placebo or the control. And it also allows for statistical test assumptions to be met. So different ways of randomizing. We're going to start by talking about simple randomization. That's the case where each participant who shows up, or each participant who's enrolling, when they meet eligibility criteria, a simple randomization is the same thing as like flipping a coin. So each of these people who shows up, we flip a coin, and they're assigned to either Group A or Group B. And simple randomization has a lot of advantages, right? It's easy to generate. It could be like flipping a coin. It's impossible for investigators to guess what the next treatment assignment will be. Even if they know, right, the first person was assigned to A, they have no idea what the second person will be assigned to. If an investigator could break the code, they might be tempted to put certain patients on preferred treatments, and that would really bias the results and be a problem for the trial. A lot of the properties of many statistical inferential procedures, like tests and estimators, are established under the simple randomization assumption. The major disadvantage of simple randomization, which you can see here on the slide, is that the number of patients assigned to the different treatments is random. So you could definitely end up with an imbalance like this. This ends up with 3 in Group B and 1 in Group A. So how can we handle that? How can we control the number of participants in each group while still actually randomizing? So that's what we call permuted block, which allows us to allocate equal number of participants to each intervention arm. So you can see here on the slide, if this is our group of participants who's going to enroll, we're going to divide them up into the first 4 are going into this block, the next 4 are going into this block, and the next 6 are going into this block. And these blocks are actually have an equal number of each arm in them, but the person randomizing doesn't know what those are. And so that way we're actually able to control. So we have numerical balance, which gives us statistical efficiency and appearance of success. We avoid bias in the secular trend of patient characteristics and staff implementing protocol. We have balance or near balance if recruitment is stopped prematurely because we've got our blocks. And it's a further safeguard against bias. So we also, in addition to having these blocks, then the block sizes are also random. So the person randomizing truly cannot predict what the next participant is going to be randomized to. If block size is known and the intervention is not perfectly masked, once previous allocations within blocks are done, then the last allocation would be discoverable. So that's why we want to differentiate the block size. So you can select the block size in random order. And so just to summarize, this is a two-step process. So you randomly select the block size, and then within each block, you randomly select the order set for the groups. And then the next issue is that we might want to be able to control a little bit more about our participants. And that's where we talk about stratified randomization, where we want to control a known confounder. And so here, suppose we have 4 green people and 4 orange people, and the color of these people is a known confounder to our randomized trial. Then we want to be able to make sure that we're controlling that confounder within our treatment groups. And so therefore we're going to randomize, we're going to randomize each of these into our arms separately. So whatever randomization scheme, here I'm showing simple randomization, but more than likely it would be the permuted block. We would do each of those randomization schemes separately for each kind of person based on that known confounder. And so this makes the treatment groups appear similar. It can give more credibility to the results of our study. Stratified sample could yield more precise estimates of treatment differences, but one must be careful to conduct appropriate analyses. When one or more or a few baseline characteristics are strongly related to the outcome, that's when you want to use stratified randomization. So in the Harvest trial, for example, we used a permuted block randomization algorithm of different sized blocks with a 1-to-1 randomization ratio to the 2 trial arms. And because we knew that we had 4 sites in the Harvest trial, we knew that site would be really heavily affect our outcomes. We also knew that HIV status, positive or negative, would really affect our outcomes. And we knew that the tuberculous meningitis grade 1 or 2 to 3 would also be a confounder. So we wanted to stratify by each of those. So the randomization process was that each clinical site had 4 separate randomization schedules prepared based on HIV status and the TBM severity. Now let's talk about group randomization. That was we just talked about randomizing individuals. But sometimes a study is designed to handle groups or communities. And in that case, we want to randomize, we do clustered randomization. So the unit of randomization might be a clinic, it might be a community, it might be a family. Here I've represented, like a school. Maybe this school has 3 people who are going to participate, this school has 6, etc. And in that case, each of these communities is going to be randomized to one of the intervention arms. So the unit of randomization is the cluster, the community, the school, but the unit of analysis is the patient. And so we want to keep in mind when we're conducting our analyses that the patients within clinics, the students within a school, the family members within a family are all going to be correlated. And so we understand that when we randomize, and we understand that when we analyze. So we're going to deal with variance both within and across clinics, families, communities. Again, when you consider you would, even in this case, consider stratification. Maybe you have a group of schools, you want like high population schools. Maybe the size of the school is going to be a confounder for your outcome, in which case you would randomize your clusters based on that stratum. Maybe it's a urban versus rural clinics that you are randomizing based on that strata. So that's something to keep in mind. And so that's kind of my overview of randomization. Before I move on to blinding and masking, are there any questions about randomization?
Ma'am, in one of the articles, I came across a term called ARA, Response Adaptive Randomization. Would you throw some light on that? Or do we commonly use it? Or
So
I'm going to pass this to Dr. Bangdiwala because I think he's more familiar.
Oh, I don't know what's going on here. We're going to have a whole session on adaptive, so let's not talk about it now. So that's actually this afternoon or maybe tomorrow on adaptive. So all of those we'll be talking about.
Great. Okay, so next topic is blinding and oh, sorry, another question.
Ma'am, after block randomization in Harvard study, ma'am, what you did after block randomization in Harvard study, so that the blocks have similar features.
So So in the Harvest study, we were stratifying by site and HIV status and disease grade. And then within each of those stratum, we were applying the block randomization. And so therefore, with that combined strategy, then our two treatment assignments were able to be similar across them. Does that make sense? I'm afraid I can't hear.
We have 4 different. So first we have 4 different groups like HIV, this CD4s, and then we can make blocks.
Yeah, so you have the 4 groups right there. 1, 2, 3, 4. Those are the 4 stratum. And within each stratum, you do randomization, but instead of doing simple randomization, they did block randomization within each one. So they have 4 separate schedules. The number
by the number of patients that you want to recruit in technically. If you're having a smaller sample size of 100 or 200, smaller block size would be okay. If you have a large sample size, and then if you have a smaller block size, it's easy to predict what is going to happen next. So we try to do a larger block size. So it depends on what you have on the sample size. And it's blinded information too. No one should be knowing what's the block size being used to create a randomization list. Unequal block. I mean, you apply a block to make it equal. And if you don't apply, so we apply a block size of 2, 4, 6, a maximum of 8 block size, and it just goes in random. Yes. Yeah, it goes in random. Yeah.
Okay.
yes, we can proceed.
Great, thank you. So blinding and masking procedures. Most randomized clinical trials are blinded or masked, and that means that those involved in the trial do not know which intervention each participant is receiving. Some trials are not blinded because logistically the intervention cannot be delivered in a blinded manner. In that case, the intervention arm assignment is still unknown to the study staff before randomization, right? So in the platform cryptococcal meningitis study that I described yesterday, that's an unblinded study, in part because logistically we can't make up a placebo that looks exactly like each intervention. Many of our interventions are IV antifungals. Some of them are taken orally. And so because of the logistics of the platform trial, we decided to not have that be a blinded study. All of our studies prior to this platform were blinded. We took the time to make a placebo pill that looked just like sertraline for our study that had sertraline as the intervention, for example. But regardless, the intervention arm assignment is still unknown before randomization, and that way we don't impact or bias our randomization. So why mask? Why are we masking? Because we want to maintain objectivity for patient care and for data analysis. We don't want our thoughts on how things should be to affect patient care, and we don't want it to affect data analysis, because we want the data to speak for itself. We want it to be as true as possible for good patient care in our study, and for our results to inform further patient care. So there's three things that are linked. Allocation concealment, which is preventing interfering with randomization design before someone is randomized. There's random allocation, where we're preventing interfering with the intervention effect from biased assignment. And there's masking. So that's preventing the investigators, participants, and others from interfering with the intervention effect by not complying with the protocol and adhering to the intervention. So during the trial, during study conduct, who's unaware of the intervention assignment? Pretty much everyone. The staff, the participants, clinicians, and study investigators. Many study statisticians are blinded during the study. The DSMB board, the sponsors, for sure, the sponsors distance themselves and are not aware of the intervention. Because all these people, whether they mean to have nefarious, ideas or not, or even if they want the trial to be done in an unbiased way, if they knew the study assignment for a trial that should be blinded, it could influence how they are behaving during the study. So who is aware of intervention assignment during the study? Some trials actually have an unblinded statistician who can see the treatment assignment at any time. And that they generate interim analysis reports. They are, many trials have a blinded statistician, some trials do not. And then there's the pharmacists who are going to be fulfilling the prescriptions after randomization. This is somebody who's not involved in patient care or data collection. So how do we assess who was aware of the assignment after the study ends? This is important to do just to make sure that you know if your study was at all impacted by unblinding. Was there evidence that the study staff involved in patient care was unblinded during the trial? For large enough trials, you can actually look at the baseline characteristics between the intervention arms to see if there was any unbalance, and that could be an indication that there was some sort of unblinding and maybe somebody affected things. One way of checking is to ask those involved to guess who got which treatment assignment. So we have an example here of the percent of participants in a trial of propranolol versus placebo who guessed treatment correctly. And so the people who were asked, the patients were asked, the physicians were asked, and the clinic coordinators were asked. And they had different numbers of people who thought they were taking propranolol and taking placebo. So each of these participants were asked, for each of the participants, who do you think was taking what? And you can see that it was somewhat different between them. Allocation concealment. This is done through a number of ways. Opaque, sealed, and sequentially numbered envelopes, that helps. Numbered and coded drug containers, labeled pill bottles. Central randomization, remote or local computer with an encrypted schedule, or you might create it on the spot. those are different ways of allocation concealment. Ways to keep participants blinded. So you've got your drug interventions. You want to make drugs indistinguishable, right? I mentioned that we developed a placebo that looked and smelled and tasted just as close as possible to sertraline. You also want to talk about your dosing schedule being the same, the packaging being the same. But one thing is I mean, side effects is something that you're going to be measuring, not something you can control. With behavioral interventions, it's pretty much difficult to impossible to have those be blinded. For staff and clinicians, it can be difficult in an intervention effect. Many times the clinicians and staff, they're aware of what's going on. They have access to the medical records. They're aware of what side effects different participants are having. So you want to keep intervention administration separate from participant care and management staff. In the Harvest trial, this is hard to read for you probably, but this is just straight from the protocol, the blinding and unblinding plan. The only personnel that were unblinded to the treatment assignments during the course of the study were the pharmacists, the statisticians and data managers, and the DSMB members. So for our study, we had interim DSMB reports, and we as the statisticians would generate the reports. We had an open report that was did not show treatment assignment, and then we had closed reports that showed our outcomes by treatment assignment, but we just labeled them as group A and group B. We wouldn't say like, this is the group that got sertraline, this is the group that got placebo. Just group A and group B. And the DSMB were the only people who were who were seeing this closed report. And for that, some trials, depending on the DSMB charter, the DSMB can elect to unblind themselves if it's really important based on what they're seeing in the interim report. So that brings me to the end of this unblinding or the blinding masking section. Are there any major questions about this? Or do any of the other panel members want to add anything?
No, good job.
Great.
We can proceed.
Okay. Thank you. Great. Okay. Okay. So the next section is talking about GCP principles. So good clinical practice. These are an international ethical and scientific standard for designing, conducting, and recording and reporting trials that involve participation of human subjects. These are not statutes and regulations. They do not have the force of law. Compliance with good clinical practices. What do they do? What do they do for us? They provide public assurance that the rights and safety of participants are going to be protected, and that the data and reported results are creditable and accurate. And I just want to point out at this point, one of the things that comes up a lot in clinical trials is that we're focusing both on the integrity of the way we treat humans, and we're also considering the integrity of our data. Both of those things are very important in our trials. So some of the GCP goals are to prevent problems, to detect problems, and to correct problems. And so we'll talk about those in a second. I'll highlight that the International Council for Harmonization of Technological Requirements for Pharmaceuticals for Human Use, GCP guidelines, says that the quality assurance are all those planned and systematic actions that are established to ensure that the trial is performed and the data are generated, documented, and reported in compliance with GCP. And quality control are the operational techniques and activities undertaken within the quality assurance system to verify that the requirements for quality of the trial are being fulfilled. So a quality assurance plan documents how a specific study will assure quality in study conduct and data collection. So one of these features is a data management plan. And a data management plan, it's like a formal document that outlines how data will be handled. Even if you're not going to seek funding, many times you have to draft a data management plan when you are when you're writing a grant and you're seeking funding. Even if you're not and you're just conducting research that that doesn't require funding, documenting a plan for your research data is a best practice. Also, what I've highlighted here is that a DMP is a living document. So the protocol is something that is set in stone, and if you have to update it, you get IRB approval. A data management plan, like research is all about discovery, and the process of conducting research sometimes means you need to revise something, right? So this is a document where you say, this is how I'm going to plan to handle the data. And as you go through your study, you might need to change some things. So some of the features of a data management plan are how a site is going to handle data collection, and how the data management team will query the data. Both of those things are included. Data management ethics, the goal is to maintain objectivity and maintain high quality. And so some of the elements that go into a data management plan are all the processes at the site or the data management team, training and certification, even a log to record people who have gone through the training, that's included. Supervision, checking, monitoring, those processes are written out in the plan. Documentation to make sure that we avoid possibility of tampering, and privacy and confidentiality. So from my harvest trial example, this is a flow chart that's included in the wider data management plan that shows the flow, right? So we had initial CRF case report form completion, where we're first collecting the data. Those are reviewed. So this is on paper, by hand, the data is collected. Then another independent staff, 2 independent staff are checking QC1 immediately, and then 1 to 2 days after, where they're checking, reviewing the data collection against source documents. Then it's submitted to the electronic data capture. At that point, then the file is submitted, and there's more validations and query checks. Then there's more they check the queries. Then they're sent back to the site, and the responsible staff have 2 days. That kind of timing is also included in the data management plan. 2 days to resolve queries. Then they're reported to the study coordinator. The corrected paper case report forms are then resubmitted to the electronic data capture, and the cycle continues until the data is checked and clean. So that was the data management plan. Another feature is the statistical analysis plan, which captures vital components of transparent, objective, rigorous, reproducible research. So this statistical analysis plan, or the SAP, is going to basically detail everything statistical that you plan to do with your data. It's going to include your descriptions of your analysis populations. It's going to detail, in your protocol, you had a whole reasoning behind your sample size. Your STAP might include more information about that. It's going to have details about sensitivity analyses that you have planned. It's going to have methods for how you handle missing data, etc. So the example, this is the table of contents from the harvest trial SAP. And you can see that section 1, most of these things is basically just a repeat of what's in the protocol for data, right? The purpose of the study, the study design, sample size, objectives, randomization, and blinding. Then you get into the nitty-gritty, the general statistical considerations, right? How do you define your analysis populations exactly? What strata and covariates are you going to consider? How are we going to handle missing data? What subgroups are we going to plan a priori analyses for? Then every single endpoint is specifically defined. Every single sensitivity analysis is just specifically described. All the way down even to like protocol violations. How are you specifically going to be analyzing those and presenting those? And so this is basically like instructions for how you're going to analyze your data, and then this directly translates to your reporting to the DSMB and your final reporting. It's a clear plan for analysis. And what I like to highlight is that a SAP, just like the protocol, to me, it provides a platform for the whole team to come to consensus on a plan. A lot of times in a clinical trial, there's different groups of people who are all stakeholders and invested. And when you have a document like the protocol, like the SAP, like the DMP, you are putting all of the procedures and the processes for your trial, you're documenting it on paper in a place where everyone can reference it, and your trial can go work systematically because you've got your procedures documented. So what requires quality assurance? Everything does, right? Key or critical variables, eligibility criteria, endpoints. Issues relating to compliance with the protocol that are critical to the validity of the study. Reliability of each step of generation, recording, and transfer of data. Changes and clarifications to the protocol. Documentation of ethical considerations, the consent process, IRB approvals, etc. So like I've said earlier, there's those 3 quality assurance elements: prevention, detection, correction. So some of the ways that we prevent any issues before we even get started is a really well-written protocol, a good manual of operations, a collection limit that's limited to essential items, right? We want simple. Simple keeps us, simple is not complicated, right? Simple means that we're going to have higher quality stuff. We're going to have less chance of error. You're going to pre-test your study forms. You're going to design them, and then you're going to test them out so that you have more input, so that you have everyone's on board with what the case report forms are collecting, and everyone is aware of the definitions. Investigators' commitment to follow the protocol. Everyone needs to buy in. This is like what I was saying before. When you document things in the protocol, when you document your data management plan, those documents provide the platform for the team to have consensus about how the study will run. Training and certification, that prevents quality issues. Centralized processes, like data from a central classification committees, calibrated equipment, these kinds of processes make sure that we have quality data. And then maintaining study records, audit trails, and archiving. So that's prevention. That's getting things, that's making sure we don't have error quality issues. But if some things are going to come up, so we also need detection, right? So a central monitoring of the data on individual patients, data entry system checks, logical consistency checks. These are things that the data management team will program. Site visits, standard checklist, records audits, comprehensive performance monitoring reports, etc. You'll have internal monitoring and external monitoring a lot of times. And then statistical investigation into the aggregate data by site and by staff. And that lets you identify unusual patterns, lack of variability, unusual relationships in the data. All these things are for detection. These are things that are ongoing during the trial to make sure that you're catching quality issues in real time. And then when you detect them, you need to be able to correct them. You need to be able to correct the errors, minimize the chance of future occurrences. You want procedures to be implemented early so that you're catching them early and you're not dealing with a bunch of errors throughout the study. You want to empower individuals, committees, and centers to address problems. This will have an effect on systematic errors, bias, violations of protocol. You want to address individual site or staff performance, redress misconduct or fraud. In our studies, I'll show you a little bit later how we collect on paper case report forms that then get submitted to the electronic data capture. And so we have young medical officers in Uganda who are collecting the data, and sometimes they rush, sometimes they don't collect things right. And so one of the things we implemented was they were assigned a researcher code, so a staff code. So on the case report form, they had to sign it with an illegible, doctor signature, but they also had to record their staff number. And so if we were finding a lot of issues with this, a lot of errors in the data, we could see how many errors were popping up from a specific staff code And that way we could see, is it a one specific staff member who's collecting something incorrectly? We can address that. That's just an example. So there's clinical monitoring versus data management. This is what I was saying again before, where your quality assurance is both for your management of your staff and investigators, and your management of your subjects. Your management of your people, and it's also your management of your data. So just to highlight quality processes, you've got standardization of processes. You want to ensure that all of your processes, from participant care to data collection, are conducted the same way for all participants throughout the trial. That is a benefit for the participants, and it is a benefit for your data. The standardization of processes also serves as a guide for trial staff, which is especially important for studies that enroll for years, right? You have documentation of your processes that veteran staff can reference if they forget, that new staff who come on can join, review the processes, and pick up right in and maintain that consistency, which, this ensures consistent data collection. I will also say that the standardization of processes, they can and should vary by site, because sites are generally a randomization stratification factor. Like I mentioned for the Harvest study, we had 4 sites that each had their own randomization schedules. And so we weren't as worried about the South Africa site, for example. They were able to have their own processes in place without having to be concerned about matching exactly what was done in Uganda, because those sites are so different. That also gives each site, they feel empowered to create their own processes that work for them. And that's important, because you need staff to feel empowered and to have buy-in. Speaking of staff, you also have training and certification of staff. And so that ensures accurate and consistent conduct among trial staff. And I will also say that the bonus is that there's team camaraderie. The more trainings that happen, I think the more people buy in, feel empowered, and then they also feel connected to the team. I should have put a picture here of many of our trial team photos, but I'm sure you've seen them with other studies you may have been privy to. But when you get that team photo, that team camaraderie, it happens often at either trainings at the beginning of the trial, or of course at the study end, at the unblinding, at the where everybody congratulates themselves for working on so hard. But it's really important at the beginning, everybody comes together and gets trained, because it helps with study conduct, honestly. So this is an example, like I just said, in the Harvest trial, we had a Ugandan site, we had South Africa site, we also had 2 sites in Indonesia, and each of those sites operates differently. And so this is an example of all of the standard operating procedures, the SOPs for the South Africa site. They had some SOPs from the sponsor. They had some SOPs that were study-wide, right? The data safety and management plan, that DMP, that was the same for all sites. The way they recorded adverse events and reported them, that was the same for all sites. The way they managed antiretroviral therapy, that was the same for all sites. The way they managed DILI, which is a liver injury, that was the same. Randomization, while they each had their own schedule, the way they conducted randomization was the same for all sites. But then down this lower section here is site-specific SOPs, right? So the study, the overarching study only specified 6 SOPs, and then the sites were empowered to create their own way of documenting of medical history and the physical exam, conducting those, their own way of HIV testing, their own way of collecting blood specimens and storing them, those kinds of things. They could each site could determine their own procedures, and they still needed to document them and share them with the overall team, so that, again, documenting it, making everyone aware, references, everyone's on the same page. And one other thing with quality processes is you've got calibration of equipment and laboratory QA. This should happen frequently throughout the trial. This assures that equipment will measure accurately, will measure the same manner for all participants. And it's also important for consistent patient care and data, which is again, those two things that go on in a trial. We have to be aware of how we're treating our humans and how we're treating our data. And then I'll move on to data processes next, but I'll just ask if there are any questions for quality processes, quality assurance, GCP, or if any of our other panelists want to add anything.
Good morning. I had a question. So this is quite detailed, and I don't have any prior experience with writing a trial protocol, and it would be very helpful if you could sort of direct me towards some reference generic protocol or something, some such resource which is available, so that we don't miss out on these key points in our protocols.
Yeah. So, I mean, I can mention that there are templates for protocols. There are templates for data management plans. There are templates for DSMB charters. There are templates for the quality assurance plans. All of these, there are templates for these. Unfortunately, those templates, if they're developed by a pharma company, they are proprietary. If they're developed by a research institute like our research institute, they're intellectual property. So we tend not to share those.
Yes.
However, you can find online some of these things because some people may have published it. A good place to look for them is in the trial registries because people will have to register their protocol. Whether it's at the ICMR one or at the WHO one or the US NIH one, you have to register your protocol. So you can take a look at those. That's one place to look at it. You can also look at, some people will put it in a publication, but that's more rare. I think the best place to look for these things would be, at least for the protocol, they will be in the those registries.
Because
the clinical trials,
registry.
It's mandatory to do it.
Yes. But it's mandatory to register them, but whether they'll be openly accessible,
Yeah.
I mean, so I'll definitely have a look at it, because it entails a lot of imagination of all the possible scenarios, and before one has even recruited the staff or even gotten the funding for it, so there is a chance to miss out.
Yeah. I mean, there's a lot of, details of so many things. The protocol itself will not have so many of those details, right? They will have a general idea. Yes, we're going to do central laboratory analysis, but they will not say necessarily what is the quality processes and the calibration, how they're going to do it in detail. If they ever publish a paper on that, you may find it there, but usually you will not find that there. The manuals of operations, these quality assurance or quality assurance plan, or this, especially the statistical analysis plan, you're not going to find that. People are not going to publish that. They will publish in the main paper. They will have a little section on the methods of that, but they don't have all the details of that you will have on them. That's very hard to find in general.
I will say that yes, I will say that there are some folks who have tried to publish like general templates that could be available. Like if you search PubMed, like somebody has tried to publish like this is generally what you would want in a SAP, but they would include everything and you have to tailor it.
Regarding this site-specific SOPs, do we have to have all these SOPs while we submit to the ethics committee? Because when it is multi-centric, maybe the ethics committee clearance happens at different points. So usually one place gets it and then the subsequent place gets.
Yeah.
So usually the other places will be just developing the site-specific SOPs or something. So is it necessary that we have the site-specific SOPs in the master protocol?
Yeah,
when
it really
is submitted
depends on
first?
what your ethics board requires, right? It's not, you cannot give a blanket statement. If you have them, it's best to share them. Now, SOPs in general are needed. It's ideal to have standard operating procedures. They are required if you're going to be doing a study that is subject to a regulatory agency. So the regulatory agency will demand that you have standard operating procedures for certain processes. But if you are doing a trial that is not subject to a regulatory agency, so you're doing an educational one, or you're doing a, one that is, we've done environmental interventions. These sorts of things are not going to go to a regulatory agency. Yes, you should have standard operating procedures, but they don't have to be as rigorous and detailed and specific of this is how we're going to measure this is how we're going to do that, this is how we will validate this, etc. All of those details are not necessarily required. But whether you submit it to an IRB or not, that depends on the IRB. And every IRB tends to be very different. Yeah.
Great. I will Will I continue with the last section?
1 second.
Can proceed, ma'am.
Okay, great. Thank you. Great questions, everyone. Finally, I want to talk about a couple of features with data processes and systems. So the first, we've talked a bit about data at this point, and data capture is essential in your randomized trial. You're conducting this so you can collect data, so you can analyze it, so you can answer your research question. And so you are, for your study, you're going to design case report forms. These are the forms, this is the where you're going to document each of the things that you're going to measure throughout your study. And so the way these are designed is unique to your study. It's based on your study aims and your endpoints. It also is important to involve everyone who's going to be involved with your study, especially your clinicians and your data management and statistics team, because you all want to be on board that the way you design them works for both in the clinic and with the way you're going to manage the data after it's been collected. So first, this is an example of a schedule of events that was detailed in a protocol. The clinician who drafted the protocol was aware that we're going to want to collect data on screening. We're going to want to collect data on a lumbar puncture, past medical history, the TB history, HIV status, all of these things. We're going to also, because this particular trial, the endpoint was survival, we're definitely going to want to document vital status. We're going to want to do the cerebrospinal fluid. We want to collect all the data on those labs, the blood work, etc. So then what you want to think about, okay, these are all the things that we want to collect. How and when do we want to collect them? And so basically you plan your trial to have specific visits, scheduled visits. And some trials might have unscheduled visits because a patient needs extra care or something, but your trial is going to have a schedule with specific visits. And each of those visits is an opportunity to collect data. And so your case report forms are going to be designed based on what you need to collect, which is on like the rows, and when you're collecting, which is these columns, these visits. And so I'll highlight here, this is the screening visit. some of the things that need to be collected at the screening visit are going to go on to a specific form, right? If you're collecting data on your cerebral spinal fluid that comes from a lumbar puncture, maybe you have a specific form that's just for those data. If you're collecting blood work, maybe you have a form that's just for blood work. Your informed consent, that's the documenting that you got informed consent, might go on its own form. But you can see here that for this trial, we were left over with a bunch of things. We needed past medical history. We needed eligibility. We needed a physical exam, etc. So those things show up on that informs how we designed our screening form. And it ended up being 4 pages long. The first page had like background, age, sex, blood point of care of your cryptococcal antigen, etc. Were they in a previous study for us? The second page was everything HIV. What's their HIV status? Are they on ART? If so, what regimen? Then page 3 was everything with their cryptococcal meningitis history. Did they have previous screening? Were they previously on antifungals, etc.? We needed to know all of that. Then the last page was their TB history, and collect what did they have prior TB? Was it pulmonary, extrapulmonary? All of those things. And so that informed us with how we designed it. And clinicians, they gave their input into what needed to be collected. And the statisticians and data management team, we gave input into how it should be designed, what kinds of fields. We needed like clear definitions of like past receipt. We know it's complete, incomplete, known or unknown, really getting into the specifics of like, what do each of these data fields, what are the definitions? Then once you have this designed, you would test it out, like I mentioned previously. You would make sure that it works before you start implementing. So this is an example that I'm showing you from a previous study of that I worked on, where we use paper forms, right? So data collection previously and even today, in many cases, it's on paper. But now we're getting into more and more that it's direct electronic data capture. So I'm going to give you a couple of examples. This is a screenshot of a software from NIH called DF Explorer. It's just DataFax because it was called DataFax because you basically recorded everything on your paper form and then you faxed it into the system. And an intelligent character recognition would review it and try and collect, it would review this, handwritten section and then enter the data into the electronic data capture. Then a human would review this to make sure that the intelligent character recognition didn't make any mistakes. And then that's how you would get your data into electronic form. Now we are transitioning into something called REDCap. I can't see everybody's hands, but can I just see a show of hands how many people have heard of REDCap or used REDCap? Okay, that looks like maybe a majority. That's great. So I think was developed at Vanderbilt University in Tennessee in the United States, and it's got a lot of features that allow for data capture in a really streamlined, easy way. It's really user-friendly for designing your forms, whether they're like repeated, whether they're surveys, etc. This is an example of a form that we're going to be using for an observational study that we're going to be doing. For our platform trial, however, regulatory reasons, we also have to capture on paper. So we have to have a source document capturing it on paper so that we can reference. Different studies, different regulatory issues. Maybe you can just do direct data capture with yours and you don't need to have anything documented on paper. It depends. I will say that regardless, having something like REDCap or even DataFax is a way to like design your form and collect your data systematically and consistently. Something you don't want to do is like rely on like Excel, right? That's probably like the easiest way to think about like a data set on the computer, but that's so prone to human error. So trials have, these are just two examples of softwares. I've worked alongside colleagues they've been on trials where they just like hire somebody to develop their own, develop a data capture, electronic data capture system specifically for that trial. So these are just some systems for data capture. Once you're collecting your data, however you're doing it, you still need edit and consistency checks, right? Even in REDCap, we have people keying it in. It's human people collecting the data every step of the way, keying it in. There's going to be mistakes. And like I said, with the quality stuff, you need to be detecting it and correcting it. So what I've shown you here is, this is an example of somebody entering data directly into REDCap, and they tried to put a white blood cell count of 99.9. And REDCap, you are able to program an alert that stops somebody right then and says, hey, this is out of the range that we're expecting. If it's true, leave it, but you probably want to double check it. So that kind of thing can happen live in real time. There are other queries that maybe need to go across forms where, we're checking things like, okay, you told me on the day 14 visit, like this person had on the day 14 visit form, you told me that there was an adverse event, but we have not received the adverse event log into the system yet. So it's that kind of thing that like alerts and checks people. So in our current platform trial, we have these queries running every night, and the sites can go in and look at, okay, what is not complete in the data? What is potentially inaccurate? Like here you're seeing there's potentially a closing pressure for a lumbar puncture that's maybe it's out of the expected range. We need to double check it, that kind of thing. So these edit and consistency checks, they either happen live right at data collection for an electronic data capture, or they happen later if it's the kind of system either where you're collecting on paper and then you need to enter it later, or if it's something that happens across forms and the data management team needs to program it to detect. Regardless, like I said with quality checks earlier, we set these things up as best we can before the study starts. We program these things understanding how we want to collect the data before the study starts. And then when the study starts, we are proactively checking things to make sure that we're catching any issues early on, so that we capture the issues early on. We catch them, we make the corrections, and quality of data improves. Okay, so this next thing I'm going to talk about is CDISC, which, or let me see a show of hands again. Who has heard of CDISC compliance? Okay, so CDISC is something, apparently it was started in the late 90s. I did not know, but now, almost 30 years later, it's really starting to take off and be required among a lot of trials. It's basically, the goal is to standardize the way data is collected and handled in order to improve quality and also interoperability of data in medical research and healthcare. So it's a set of standards that tries to help all data collected be sort of in the same format so that it can be easily understood outside of your trial. It can be shared and validated, and et cetera. So within CDISC, the Clinical Data Interchange Standards Consortium, you have a number of standards, and 3 of those are C-, which is a set of standards that sort of helps you decide what your field names on your CRF are going to be. So all of these little elements on this CRF are called a field. And then on the back end, you've got a variable name. And so C- is going to tell you, okay, for opening pressure on the lumbar puncture form, that's going to have, that field should have this specific name. Then SDTM, once you get from collecting the data with CDASH, SDTM is a standard way of sort of categorizing the data sets into domains. Like this is all background data. This is all laboratory data. This is all outcome data. And then ADAM is the standardized way of then compiling your analysis data set, including any derived data. Like you took these fields from this and used them to derive a new field that you used in your analysis. ADAM has standards for ways you are organizing that data. And finally, the last thing I'll talk about is when you finish your study, you are going to work really hard to have clean data. You're going to work hard during the course of the study to have clean data. But when you get to the end, there's going to be a push to where you make sure you have the least amount of missing data possible. Outliers are accounted for and understood. Everything is clean. And when you get to that point, then you're going to lock the data. And it's when you get it clean and you have it ready for analysis, you have your final results. You lock the data set, and that's the individual data set prepared for publication. And then the data freeze is the comprehensive database of all those locked data sets that have been received or prepared by a study prior to any associated publications being published. So there's become this new, these requirements that if you're going to publish, which you should, when you publish, you have to have your data ready to be shared at the same time so that there's transparency, reproducibility. And with that, I think that's my last slide. And I will ask if there are questions or if any of the panelists would like to add anything.